you back for this learning session of biosensors. In earlier sessions you have learnt about various components of biosensors, what is a biosensor, and various components present in that which led to classification of bioreceptor based biosensors as well as transducer based biosensors. So as I told you already, this couple of videos will be exclusively for bioreceptor based, and in that bioreceptor also already you have seen biocatalysis based biosensor, which is exclusively based on immobilizing enzyme. If you design a biosensor, we call it as biocatalysis based biosensor, right? So coming to the next topic in this series, that is bio-affinity based biosensor. So just to remind you, what is affinity? Because straight away instead of telling about bio-affinity or how a bio-affinity based biosensor is designed, you should always think, what is affinity? Affinity is nothing but attraction. So do you find attraction within the molecules? Or if I say bio-affinity, do you find attraction with biological molecules? That is how the main difference between affinity and bio-affinity. So all these questions to be answered in your mind before we enter into this topic, right? So for example, what is affinity? What is attraction? Always positive charged ions attract negative charged. So there is an affinity. So in biological molecules also, if you see, there are certain biological molecules which attract each other. For example, you take an antibody which is attracting to a specific antigen and binding, right? Similarly, you take DNA. DNA is a double-stranded molecule, but once you break it, you have two single strands. But these single strands will only complement each other and they bind. Why? Instead of going till DNA, you can even think about these nucleotides itself. So if you take in general, like instead of going for all these things, if you just imagine always a G attracts C, right? Always A attracts T. Never ever G binds with T or A binds with C. This will not happen, right? So this kind of attraction which you find within the molecules is called as bio-affinity, right? Then comes the next question. If something is attracting, is that alone is enough for us to design a bio-affinity based biosensor? If it is attracting everything, for example, you take a magnet. Magnet usually attracts iron. But is there any magnet which only attracts needle shaped iron or rod shaped iron or some form of iron? Can a magnet, while attracting and while attaching, can it differentiate? Is it possible? Because that kind of exclusive way of attracting is required for designing a biosensor. So as I already told, as we already discussed, in biological molecules there is a lot of attracting molecules. Always these charging molecules start attracting one another. Instead of straight away going towards that, you need to identify a specific binding things, right? So this kind of principle becomes the basis for designing any kind of bio-affinity based biosensors, right? So I'm giving you a simple example. As I already told you, there are various types, out of which antibodies. So here while designing, again you should remember, just like how we have discussed in biocatalysis based biosensor, whether a substrate has to be used as a biological recognition element or an enzyme must be used. This kind of confusion should never ever come in your mind, right? Always you have to choose an enzyme. Substrate must be in the sample. So this sample has to have an affinity, and because of this affinity there should be some reaction. That reaction need to be converted by transducer, then that output you should understand. Similarly, when you start designing bio-affinity based biosensors, always if you want to go for antigen antibody based receptors, then remember antibodies must be immobilized on your transducer or the platform whichever you use to design your biosensor. Similarly, if you are going for single stranded DNA, then there is no question of which should be used. Complementary DNA can be used, and you are expecting a DNA which is available in the sample and it has an attraction that can be separated. Then receptors, usually always you need not have a specific receptor. Antigen, antibody is known to you, single stranded DNA is known to you. Where will you use receptors? Most of the times this comes during identification of an hormone. And here you should also remember one more point. Hormones need not have only the receptors that are available. Even a hormone can be detected using antibodies also. But to make it more precise, to make it more accurate, current generation bioaffinity based biosensors, you are getting receptors that are immobilized, right? The principle, everything is same. Basically, you have a platform wherein your biological molecule, in this case, nothing but antibody is immobilized. So when you expose with analyte, just imagine these are different types of analyte. When a specific antigen comes and binds with your antigen, comes and binds with your antibody, this association causes a reaction. This reaction is understood by transducer. I am reminding you once again, transducer is nothing but a device which converts one form of energy into another form of energy. And in our case, when you go for a biosensor, most of the times transducer converts a mechanical energy into an electrical energy. Any form of energy like light energy, sound energy, whatever input it is, in our case mostly it becomes the electrical output. That is how the processing unit processes the information, and if requires it even amplifies the information, and the data is transferred and it is displayed. So once it is displayed, it is easy to understand. So always a display is nothing but a readable output. You should understand. It can be in the form of digits, or it can be in the form of color, or it can be in the form of words. It depends, it varies. In latest you are seeing even words are also coming, like it is mentioning what stage it is, what level it is. All this you are able to see, but all this happens in the process of converting or amplifying by microprocessor and all these things. But we are not touching that part. Our whole interest in this video is about only receptors, receptor molecules. So let us focus on how a antigen and antibody binds, or how a single stranded DNA and a double stranded DNA attaches with and make a complementation there, and how that principle is being used. And for your convenience, instead of discussing all these aspects in the same video, I have divided it into two parts. In this I will be focusing majorly on antibodies and receptors, that is hormone based and antigen based, and DNA I will be giving you an exclusive video in the next one, right? So let's come back to our own story. So where do you get these antibodies? So if you want to design a biosensor basing the principle of bioaffinity, as we already discussed, we have to immobilize an antibody. And I hope you remember the structure of antibody, which is shown here. So basically it is a Y-shaped structure which is available. So in this, if you see the number one part, this part we call it as variable region, and the second part, number two, is called as constant region, because there are thousands, in fact there are so many antibodies, and majority of the antibodies have the same structure. Then how one antibody is different from another antibody, if the question comes, always look into the aspect of variable region, that is 1. The constant region is same. So the specificity of an antibody is decided based on the presence or the differentiation that you find in the variable region, right? So again, if you see the third, heavy chain. Number 3, this part is considered as heavy chain region, and next number 4, this is considered as light chain region, which you see here. So if you want to classify again, instead of going for variable region and constant region, you can even separate them as heavy chain region and light chain region. So the main recognition site, number 5, if you see here, this is the recognition area. So out of the whole variable region, this particular part actually makes the difference, right? So this variable region is specific to a specific antigen. So when you want to select a specific antigen, the difference you need to look into this part is this area. Suitable antibody has to be selected, and that antibody only you are supposed to immobilize. Then comes the next question. Okay, now you know which kind of antigen you are looking into. You want to isolate, or you want to select, or you want to detect the presence of that particular antigen, and you want to take an antibody. Then where do you get these antibodies? Are there any further types? If you see, it can be classified into 2 types. The first one is monoclonal antibody. The first category is monoclonal antibody. Whereas coming to the next part, that is polyclonal antibody. I hope you all know the differentiation between monoclonal and polyclonal antibody. The major difference, if you see here, antibodies produced by a progeny. So the generation which you get from a single parent B cell. The main difference comes here. If it is from a single type of B cell, we call it as monoclonal. Or when it comes to polyclonal, different B cells produce antibodies against the same antigen. So in both the cases, you should remember the antibody is specific for an antigen. But from a same type of B cell or from a different type of B cells is the actual scenario. If it is from the same type, then we call them as monoclonal antibodies. If it is from the different B cells, then we call it as polyclonal antibodies. So mixture, basically what it is nothing but polyclonal antibodies are a mixture of monoclonal antibodies. All this decides and depends on what kind of differentiation or what kind of diversity you find within your antigen. If it is highly diverse, if it is having a lot of variants within it, when you want to check in a lot of a big mixture, then it is always better to go for polyclonal. If not, if you want to be more specific, more precise in selecting your particular antigen, then it is better to go with monoclonal antibodies, right? So this is the general information that you need to remember. So now you understood, I hope, what is an bioaffinity based biosensor. Within this bioaffinity based biosensor also, what kind of benefit you get when you start using antibodies as a biological recognition element. So let me give you one popular example. Here, if you see, mostly you must have come across this urine based pregnancy strips. So I think you must have seen this lot of advertisements or so many places where people used to confirm whether a person is pregnant or not. So basically, how does it work? And on what basis this single strip is deciding whether the person is a pregnant or not? What kind of analyte is present? So you can already see we are collecting urine. The sample is urine. So in general, let's forget about whether it is pregnancy test or something else. In a urine sample, what are you finding? Mostly it is meant for removing of your nitrogenous waste in your body, right? Since we are ureotelic animals, if you take any human urine sample, it should contain urea. Urea is sent, right? Urea is excreted. Then along with urea, if there is any other molecules which are available inside our body because of our whatever biological metabolic activities happen, there might be some or other metabolites which must be releasing along with that. For example, you take a diabetic patient. In diabetic patient, you see presence of sugar, excess sugar. For example, if somebody is suffering with some bacterial disease, sometimes you might even find presence of some bacteria when you go for urine sample cultures and all. or sometimes you might see presence of other metabolites which are harmful for the body. So most of the times that is why when you are sick and a doctor is unable to diagnose just by seeing your symptoms, they suggest you to go for any urine sample or blood sample analysis. So that kind of information you can get from this. So in urine sample, if a person is pregnant, if a lady is pregnant, what you find is this HCG. This is nothing but human chorionic gonadotrophic hormone, which actually helps the pregnant lady during pregnancy. It helps to develop the placenta. So placenta, I hope you all know, it's the connecting link between the mother and child for supply of nutrients and what all required for the baby, right? So one general understanding is the HCG levels gradually increases from the day of pregnancy or from the day of fertilization till the date keep on increasing. So the number of days increasing, the level of HCG is also increasing. So what happens now? So you take urine sample. This is the basically the strip that you use. I mean the person who is checking for the pregnancy can use this. So when you apply sample, that is nothing but urine sample. When you apply 2 or 3 drops of urine sample, this strip basically has 3 lines. Or for your understanding, imagine as 3 layers. Okay, let's make this layer 1, layer 2, layer 3, right? So this layer 1, you remember it as a reaction layer, because this is where the sample and our respective antibodies are reacting with one another. That is why I am calling it as reaction layer. Next, second layer which you see here is the test layer or working layer. That is where, because of this interaction, whatever, whether it is a positive or negative, all this work happens in the second layer, right? And the third one is the control layer or reference layer. See, there is a strip available. Whether it is working or not also, it is very much important for us to identify. It's more than important to find whether she is pregnant or not, right? So to confirm this, you have a control line also. So now let us go into the principle. What happens? Near reaction layer, you have a antibody which is freely floating, and this antibody is not alone. It is also linked with an enzyme called helicase. It is also linked with an enzyme called helicase. So when a sample is added, if there is HCG, human chorionic gonadotrophic hormone, available in the sample, that binds with this freely floating antibodies, which is linked also with an enzyme, because a antigen antibody reaction cannot be understood by naked eye. You need some kind of visualization, some kind of understanding. That is why a enzyme which can give a color reaction is also linked with this antibody. So after their association, after the HCG hormone association with antibody, these start moving towards the second layer where actual test happens. Here there is one more second antibody which is fixed, which is immobilized. Here you have a freely moving antibody, but in the second layer you have a fixed antibody. So if HCG is linked with these freely floating antibodies, and when they come and attach with already immobilized antibody, so here you can see antibody 1, antibody 2, and in between you have HCG hormone also. This kind of condition we call it as sandwich, because the molecule is being sandwiched by 2 antibodies which are already available within the strip, right? So if the sample other than HCG, there will be so many other proteins, there might be so many other molecules also that are present in the particular urine sample. So some might be, they will not attach with this second molecule, second antibody, because they are not so exclusive for the HCG, right? that is when they move on. So when they move on, they go and bind with the third. That is nothing but the control area. So if the person is a pregnant and if her urine sample contains enough quantity of HCG, the binding happens at second layer. Irrespective of HCG, whatever is there, along with HCG also, the binding happens at third layer. So, based on this, you can easily understand. Here you are seeing one line. Here you are seeing one line, but here you are seeing two lines. That means, if a person is pregnant and if she uses this particular strip, if the strip is showing two lines, wherein that is nothing but the test line as well as the control line, both showing pink color because of the helicase enzyme that is already attached to the antibodies, then you can confirm she is pregnant. Instead of two lines, if there is only one line, that means the test line sandwich did not happen. That is why there is no coloration. I am reminding you once again, the antibodies that are present on the test line are so exclusive for HCG. That is why the binding must happen there. Sometimes there might be problem. For example, it is too early to detect. She is having only 2 or 3 weeks after fertilization, means it might be a difficult thing to confirm because the HCG levels are too low. That is why you need to wait for couple of days. So after second month or third month, if you check, then what happens? If she is really pregnant, then you will be finding enough quantity of HCG within the sample. Then obviously what happens? The binding happens. So the control line helps. So from this diagram, again I am reminding you, the control line helps you to confirm whether the strip is functional or not. And the test line confirms whether the person is pregnant or not. In both these cases, there are already immobilized antibodies which will not move from there. They are fixed in that particular place. Whereas in reaction area, there are freely moving antibodies present. So your antigen is HCG. HCG. If this HCG is available in this urine sample, if not any other is there also, this freely moving antibodies will capture them. If there is any HCG available in the analyte, then the test line captures them. That is why you get a coloration because of helicase enzyme. If there is no HCG, then control line gives you the color. That confirms you the working of the strip. So this is how basically a biosensor works. So here comes the confusion, or here you might get a doubt. As I told you in earlier classes, biosensor should contain various components like a biological recognition element, a transducer, a microprocessor and a display unit. Mostly these 5 plays a key role, right? But here if you see, you have your antibodies which are acting as biological recognition element. Then the transducer part, if you see, it has to convert one form of energy, where here if you see, instead of converting into electrical signal, it is giving you coloration. So actually speaking, the role of transducer is not available because the helicase enzyme itself is doing the part. So this is the early phases. Today, if you see in market, there are a lot of biosensors which if you remember, in biocatalysis based biosensor also I told you dipstick biosensors. This you can consider as a dipstick biosensor. So to this extension, what you have seen so far is the basic science and the principle behind designing a biosensor. So if you see the same thing, the same concept, there is no change in the concept, but you have a biosensor which gives you digital output. So as I told you in the earlier itself, nowadays instead of just giving you number of signal kind of thing or just number kind of thing, it is even showing you this display device gives you whether the person is pregnant or not. She is not pregnant. If she is pregnant, which month? This kind of clarity is coming. So you can see the whole device. I am giving you the inside model also, how it looks like. So if you see, there are various parts. The first part is absorption strip. If you remember, the absorption strip is nothing but the analyte has to be absorbed. Because if you add a urine sample, it should not move away, it has to be absorbed. So once absorption happens, then there is another part which is nothing but the battery. Because now you have a device which has to function, it has to give you display, it cannot give it on its own. In earlier dipstick methodology, you need not require any kind of battery because there is a enzyme which is giving you color. But this is the next version of it. This is the actual biosensor we are speaking. So you definitely require a battery. And the third important part is immunoassay strip coated with monoclonal antibodies. So this is our biological recognition unit. Right? Earlier absorption strip is only absorbing the sample. After absorption, what should happen? They should bind with your biological recognition element. In this case, it is monoclonal antibodies. Monoclonal antibodies should attract them. And because of that affinity, the binding should happen. Once binding happens, then the rest of the things. So you have a LCD screen, which you can see from this LCD screen. In LCD screen, this is how you get the display. But straight away, you don't get any display. After reaction, there are a couple of things which should happen. Then you have a PCB, you have a microprocessor which carries out like spectrophotometer kind of thing. So once monoclonal antibodies binds with this antigen, whatever is happening has to be recorded. That part is taken care by the spectrophotometer, that is nothing but microprocessor. Then you have 3 LEDs, nothing but they measure the absorbance and reference, control and testing zone. Here also you have all 3 zones. The reference area, the control area and the testing zone. So once the binding happens, based on that they will recognize it. Then there is a photodiode. Here in this case, the photodiode acts as a transducer. So from the term itself you can understand basically light. It is understanding the light, intensity of light. Then photodiode is nothing but a semiconductor device. So semiconductors are also one of the mostly used transducer thing, which gives you an electrical impulse, and then CCD you have for the conversion part. So all these components linked with one another, and whatever earlier you got, in the earlier example also you have seen how you are getting the device. Whether if there are 2 strips, colorful strips, then we can tell she is pregnant. If it is only one, the strip is working, but she is not pregnant. Similarly, in this kind of biosensor, there will not be any further confusion. Straight away we are telling. Straight away you get the display, whether she is pregnant or not. Whether she is pregnant or not, right? So this kind of benefit you get when it comes for regular. This kind of benefit you get when you use these kind of biosensors. There will not be any further confusion also, including how many months, what stage she is, everything you are getting. So let's see the difference. Precise, very precise information you get when you use biosensors, right? So here I am giving you one more example. Earlier you have seen one popular example. Like this there are several examples. Right? So in this example, if you observe, basically here I think the image is not clear, but let me explain for you. Basically it is checking for presence of a bacteria. Most of the times our antigen is either a bacteria, a virus, even a virion. All these things can be considered as antigen. So if somebody is suffering, somebody is sick, if you use a bioaffinity based biosensor, wherein the specific antigen is coated to a transducer, or if a specific antigen is immobilized to a transducer, that can actually detect. So once detection is done, then what happens? Easily you can recognize what kind of problem that particular person is facing, right? Like this I have given you. Like this I have given you the list of various antigens like this kind of disease, if a person is suffering, then what kind of antibody is used, what kind of transducer is used, and more important, I am telling you, accuracy and even limit of detection, how less sample is enough to recognize, and how fast detection time. Here if you see, less than 2 hours. Here if you see, 8 minutes, 10 minutes. Today if you see, there are so many biosensors which is giving you within less than 30 seconds, less than 10 seconds. But still, lot of development is going on. I have given you a couple of references which you can go through. But the principle is same. Whatever I have explained you for the pregnancy test, the same principle is applied in every way. Only difference comes is what is your antigen. For that antigen, what kind of antibody is used. This actually makes the difference, right? So you can see all these various categories, and there is the next category. As I already told you, bioaffinity based biosensors, there are 3 major groups. Either you can use antigen antibody, or you can use hormone and hormone receptor. Earlier also I have shown you a hormone binding with antibody. In this example I will be showing you a hormone binding with a hormone receptor. Right? So where do you find these hormone receptors inside the cell? Inside the cell plasma membrane, there are several receptors which will only allow a specific molecule. So a receptor which allows hormone is considered as hormone receptor. That is the basic example if you see the whole diagram, right? So here if you see, I have taken a specific example which is published in this article. So if you see, there are 3 parts here. Each part is explained. So the first one is, this is the first part, that is biotinylated peptide. Then agonistic molecule, the small molecule, and then antagonistic molecule, this molecule, right? So here I have taken the example of endocrine disrupting compounds. See, most of the times people use a lot of pesticides in agriculture fields and all. All these things are again coming into the water bodies. The excessive applying of all these chemicals, all these pesticides, what happens end of the day? They come into water streams. And once they come into water body, they are entering into the food chain, which is causing a lot of problems for the, either it may be aquatic animals, or if when those aquatic animals are consumed by some person, then even that person is also affected, right? So if you can detect the whole process, then it is good. From the term itself, you can understand endocrine disrupting compounds. That means if these compounds are exposed to our body, or if they are entered inside our body, then what happens? They are going to destroy the endocrine system. They will target one specific hormone, or they may target all hormones, which will lead to a lot of problems. So here if you see, label-free surface plasma resonance. This is SPR is one of the popular technique which you will be learning in next videos. I will be telling exclusive when we talk about transducer. But let us focus on the biological part here. So to detect specific EDC, this is a 17 beta estroidal. This is one kind of endocrine disrupting molecule. Today we are going to check whether its availability or its presence in the sample can be detected or not, right? So if you see here, this biorecognition element is combination of sensitive peptides. So when their amplification agent, what happens? So when this estrogen agonist, antagonistic binding and agonistic binding, you can see from the diagram. So if this presence is there, if binding happens, immediately what happens? That binding is showing you a response. The transducer understands. If binding is not happened, then you get a response. So when these molecules are coated on a transducer, right? When a particular molecule is coated on a transducer, if this compound comes into the picture, or if this compound is exposed to that biological molecule, that binding is causing a signal. That signal is understood by the transducer and immediately is transferred. Once it is transferred, then what happens? You can easily understand whether that molecule is present or not. So this is another wonderful example to remember, right? So to conclude this particular session, bioaffinity based biosensor. So bioaffinity based biosensor basically works on the affinity, attraction. Out of this attraction there are molecules like antigen antibody, DNA, complementary DNA strands, as well as hormone and receptor, or even a hormone and antibody. So in this session I have discussed about antibodies and hormones and hormone receptors and how they are useful, right? So, and if you see how they are different from biocatalysis, whether they are good, bad, because every principle, every process has their own positive and negative ends. If you see here, these are actually fast because the attraction between antigen and antibody is so high, immediately, instantly you get, and accuracy is also so. I shown you the structure of antibody earlier, the variable part. The variable region is specific for a particular antigen. So accuracy is also high and sensitive. How much antigen is available in the sample can be easily recognized when it compares to enzyme substrate. Sometimes it may be the result might be different from what actually happened, but that is not the case with this kind of technology. Then highest possible detection. That is nothing but accurate and sensitive. Easily they can target number and large number of samples. It's not like one sample, two sample. When it comes to mostly biocatalysis based biosensor, there is a limit. Because what happens in this kind of conditions in antibody based biosensors, you can wash them and reuse. So number of times you can use it, and large samples you can use it. Even the sample size is too high also. Since the affinity is so strong, they can easily bind. But when you go for biocatalysis based, the sample is so large, means enzyme cannot handle. Such kind of issues you do not have here, right? So all this makes this particular process very easy. So I have given you a couple of references and research articles which you can go through, because there is a lot of information available, but I suggest you to go through proper and authenticated sites. Do not search everything in Google. You either search in Science Direct, where you get most authenticated information, because I am also collecting this information from reference articles, research papers and certain books. So you also follow the same. I have given you a couple of information, right? And these are model questions. You go through these questions, and while answering these questions, if you find any problem, you contact me. And in our regular class also we will discuss once again. So hope this experience has helped you, this learning experience has helped you to understand. If there is anything that you have missed or if you are not understand, please text me so that I will clarify your doubts. Good luck.